Hi guys, welcome back to my channel. So in today's video, we will study about the excretory products and their elimination. Now, starting with the excretory products. So what are the excretory products in our body and why do we need to excrete them? Okay, what are the products and why do we need to eliminate them? Why can't we keep them in the body? So usually the excretory products, the main that is harmful is the nitrogenous waste, and we need to eliminate them because it will be toxic to our body if we keep it for the long duration. So it needs to be eliminated, otherwise it will be toxic. Now the nitrogenous wastes, okay, the nitrogenous waste can be of various types in various type of animals. It can be of three types, which is more toxic. Okay, there can be other wastes also, but here we are studying only about the nitrogenous waste. So these nitrogenous waste, although we know that the nitrogen is important for the survival of a plant, but for animals nitrogen is toxic, so we need to eliminate them. But we put in plants as a fertilizer, nitrogenous fertilizer such as urea. So the first type of nitrogenous waste that can be formed in an animal, in an animal's body, is ammonia. Okay, next type can be urea, and the third type can be uric acid. Now ammonia, urea and uric acid, these wastes are of different toxicity level. So the most toxic is ammonia, and it needs a large amount of water. Okay, large amount of water for its elimination. It is the most toxic and it needs maximum amount of water for its elimination. So can you imagine a organism living in desert area which can eliminate or which will produce ammonia as a waste material? It cannot, because if it's living in a desert area, it will not have water around it to eliminate ammonia, and ammonia needs lot of water, so they cannot produce ammonia, their body will not be able to survive. So where should these type of organisms live which eliminate ammonia? They should live in aquatic habitat, right, in water region. So, most of the aquatic animals, whatever they may be, fishes, amphibians, reptiles, mammals, whatever they are, if they are aquatic, they will eliminate ammonia because very large amount of water is required. And how will this ammonia pass? Ammonia will pass through simple diffusion. Okay, through simple diffusion they will be produced and they will be eliminated within few minutes. They are not to be kept in the body for hours because it is more toxic than the other two, that is urea and uric acid. Now let's come on to urea. This is medium level toxicity. Okay, medium level toxicity, and it also requires almost a less amount of water. Okay, so we cannot say that medium level of water, but it requires less water than ammonia, but it will need more water than uric acid to be eliminated. If we have to remove urea from a body of an organism, it should require less water than ammonia but more water than uric acid. Okay, so such organisms which can live on land, they can afford to get that much amount of water. So all the terrestrial organisms, okay, all the terrestrial organisms and all the marine organisms, okay, terrestrial as well as marine organisms, because marine organisms they have salt water around them. Okay, they do not have fresh water, right? So all the organisms which are terrestrial and which are marine, they will excrete out urea. It is less toxic than ammonia. And here Kidney plays an important role. Kidney is important. In ammonia, in the case of ammonia, there is no role of kidney. No role of kidney because the ammonia will pass through diffusion. Ammonia passes through diffusion, so there is no role of kidney. But here in urea, in the case of urea, kidney is very important. And ammonia is not retained in the body because it is highly toxic, but some amount of urea can be retained in the body because urea helps in maintaining osmoregulation, osmolarity. Okay, helps in maintaining osmolarity of the body, osmolarity of the blood. So some amount of urea is retained in the body. Urea is not that much harmful that it cannot be retained. Next, coming on to uric acid. Now this uric acid is the least toxic. Okay, let me change the color. I think it's not visible. Okay, now urea is the least toxic and it will require least amount of water for excretion. So can you imagine some organisms which do not have accessibility of large amount of water, which cannot get water as per their needs? They will search for water in some areas. If they get, they will take it. If they don't, they have to search for miles and miles. Such organisms, for example, which live in aerial organisms which live in air, such as birds. Okay, such as birds, some insects, birds, some insects, snail, land snail, they move so slow they cannot get water whenever they want. Okay, some reptiles, they do not have access to water whenever they require, whenever they will need. So that is why they have such type of waste material produced in their body that will require the least amount of water. You can see, if you have observed in the case of birds, when they excrete out, their waste material is in the form of pellet or paste like. Okay, okay, so their waste material will be in the form of a pellet or paste so as to reduce the amount of water, because they cannot afford to get water, so they cannot afford to lose water out of their body. Okay, now let's start with the human excretory system. We have to study in detail in this chapter. So where is the human excretory system located? In our abdominal region. Okay, in our abdominal region. Now exact location, if we have to tell, then it is the kidneys. The kidney that we have is situated between the 12th thoracic vertebrae and 3rd lumbar vertebrae. Okay, so we have the vertebral column and it is divided into cervical, thoracic, lumbar, sacrum and coccygeal. Okay, so thoracic is 12, lumbar is 5, cervical is 7, sacrum and coccygeal is 5 and 4. Okay, so they are fused. Later they are fused. So they are fused to form one. Okay, so we can write it like this: 5 fused to form 1, 4 fused to form 1. Now between the 12th thoracic and the 3rd lumbar, our kidney is situated on both the sides, left and the right kidney. Okay. It is a bean shaped organ. We know, bean shaped organ. Color is also the same, reddish brown or dark red, we can say reddish brown color. Now the point from where the ureter originates, this is the ureter. So the point from where the ureter originates and the blood vessels enter, see, from here the blood vessel will enter. Okay, this we can say that renal artery. Okay, renal artery is entering and renal vein will also enter. So we can say like this, renal vein. Okay, renal vein will also enter. Now this portion from where everything enters, it is called as the hilum. Okay, hilum. Now this portion is called as cortical region or cortex. This portion is the cortex, and the inside this inner part is called as the medulla. So the kidney, each kidney can be roughly divided into two parts on the inner side, into cortex and medulla. The inner portion or the center or the core one is called as the medulla, and the outer region is called as the cortex. Now in the medulla, we have certain this type of structure, triangular structure, which is also called as the pyramidal structure, like this. Pyramidal structure. These are called as medullary pyramids. Medullary pyramids because they are pyramid shaped structure. Okay, inside these structures, we have our basic unit, that is nephrons. Nephrons. Okay, inside these the nephrons are situated. It will be like this actually here. Okay, let me correct. So it will be like this. Some part will be in cortex and some part will be in medulla, like this. Okay, the nephrons, when we'll study the structure of nephron, we'll come to know which part is in the cortex region, which part is in the medulla region. So these are called as the medullary pyramids. Here are the nephrons. Now these pyramids will drain their uh, collected urine, because now the urine has been formed, so they will all drain the urine like this. They will combine something like this, and they will drain the urine into a common portion that will finally unite at the hilum portion, and then it will pass on to the ureter. Now the end portion, the end portion of the medullary pyramids, okay, the end portion of the medullary pyramids are called as, so these portions, okay, end portions, these are called as the papilla, medullary papilla. These portions are called as calyces, calyx. Calyces, and this portion is the pelvis, and finally it will join at the hilum. It will go and merge at the hilum region. Hilum is the entry and exit point of the kidney, from where ureter, renal artery, renal vein will enter and exit. Okay, so this is the overall structure of a kidney. How does it looks from inside, from where the renal artery, renal vein, ureter originates, what all the basics are there. Okay, now let's study about the nephrons. Nephron is the basic unit or the basic functional unit of kidneys. Means whatever we say that kidney does this function, this is the role of kidney, whatever is done is actually done by the nephrons, not kidney. So the nephrons combinedly perform a function which is said to be done by kidney. So what is the function of kidney? Osmoregulation and urine formation. So that is done by the nephrons. Now looking at the structure of the nephrons, see, the nephron is a tubular portion. Here we will see a portion of the nephron which is called as the Malpighian tubule. Okay, which is called as the Malpighian tubule. Now, this portion, Malpighian tubule or renal corpuscle, this is made up by two parts. This one is called as glomerulus, and this is called as Bowman's capsule. This cup-shaped structure is called as the Bowman's capsule, and the red portion that you can see here is called as the glomerulus. So this, what is this red portion? It is a part of renal artery. This portion which is broader, okay, which has the broader diameter, is called as the afferent arteriole. Both are arteries, the subdivision of artery, not vein. Okay, this is the afferent arteriole, and this portion which has the narrow diameter is called the efferent arteriole. It is not a vein, it is an arteriole. Okay, it is an efferent arteriole. So the blood will come from the afferent arteriole, and this forms a tuft of capillaries. Okay, a tuft means a group of capillaries is shown here. It will form a group of capillaries which is called as the glomerulus, and then it will take away the blood, remaining blood will get away from the Bowman's capsule in the form of efferent arteriole. Now what happens when the blood comes? This blood will get filtrated through some slits or pores, okay, which is present in the Bowman's capsule. These cells are called as the podocytes. There is some gap between the cells. Through them the blood will get filtered. Blood will get filtered means the RBC will not come here. Actually, water will come, urea will come, salts in the form of ions will come, then glucose may come if the patient is diabetic, otherwise it will not come. Okay, actually glucose is filtered but it is reabsorbed. So in urine, if the glucose will come, then that means the patient is diabetic. Some amount of amino acids will come. Okay, these will later be reabsorbed. Glucose, amino acids, most amount of salt, most amount of water will later be poured back into the blood through the reabsorption method. So here in the Bowman's capsule, these things will be filtered out of the blood, except for what? Proteins will not get filtered. Okay, because proteins are macromolecules. Amino acids are the building block of protein. So amino acid can get filtered, but proteins are macro, they are larger in size, they cannot be filtered through these slits. Okay, because these slits size is smaller than the size of protein, so it will not come into the Bowman's capsule. Now whatever has come into the Bowman's capsule is now named as filtrate, and from here on we will say that filtrate is being reabsorbed, something out of filtrate has been reabsorbed, something is poured into the filtrate is moving here and there. So once the thing has come into the Bowman's capsule, it will now be called as filtrate, not urine. We will not be calling it urine here at this stage, it will only be called as filtrate. Okay, and when we talk about the amount of filtrate that is filtered, so about 180 liters per day, 180 liters of filtrate is produced per day. So not every part of filtrate comes out in the urine, because we do not urinate 180 liters per day. Okay, we urinate roughly about 1.5 to 2, depending upon the amount of water intake. If someone has taken more water, drunk more water, then they will pass out more urine, but not up to a limit that 180 liters per day. Okay, this cannot happen. So filtrate is 180 liters per day, so maximum amount of water will be reabsorbed back into the blood. Now let's talk about the overall structure of our nephron. Okay, so this is a nephron. It may be looking too much complicated, but it's very simple structure. See, here this portion is the Bowman's capsule, right? This curved portion, okay, this curved portion is called as the PCT, proximal convoluted tubule. This U-shaped portion is called as the loop of Henle or Henle's loop. This another curved portion which is at a distance from the Bowman's capsule is called DCT or the distal convoluted tubule, and this pipe-like portion, the tubular structure, is called the collecting duct. Okay, now here we have seen is the afferent arteriole and this one is the efferent arteriole. Now whatever filtrate, let's change some color, whatever filtrate has come into the Bowman's capsule will now pass on to the PCT, then it will go on to the loop of Henle. From loop of Henle it will go on to the collecting duct through DCT, and at this portion when it has entered the collecting duct we can call this filtrate as urine. So only in the collecting duct the filtrate is called as urine. Until then we cannot name it as urine, we will call it or we will name it as filtrate only. So what happens is in the PCT region, in the DCT region and loop of Henle, firstly what happens is reabsorption will take place. Once the filtrate has come, then some amount of filtrate will be reabsorbed, okay, because we have complete capillary, blood capillary is surrounding the overall all of the renal tubule and nephron. See, from here efferent artery portion is coming, it will loop over the PCT, then it will go over to the DCT. It will not come firstly to the loop of Henle, it will go over to the DCT, and from there DCT then it will come on to the loop of Henle, and here it will go and converge into the renal vein. From here it will go on to the renal vein. Now what happens is in the PCT portion whatever has to be put back into the blood will be reabsorbed. So this is called reabsorption, okay, because every portion has a blood capillary near it, so it can be reabsorbed. So this is called as the reabsorption. Okay, reabsorption means it is being absorbed again into the blood. For example, glucose has come into the filtrate, amino acids have come into the filtrate, some extra amount of water, extra amount of ions has come into the filtrate, which we do not need to eliminate, which body is requiring, which the body will need after some time, or the body needs at this time, at the present. So all of these will be reabsorbed back into the blood because they are essential. We cannot treat them like very cheap things that it should be eliminated very quickly then we'll gain it. No, we cannot do that, our body cannot afford to do that. So that is why we will reabsorb into the blood. Okay, now this is the overall structure of a nephron. Okay, you need not worry because it's not complicated at all. At first it may look complicated, but after some amount of revisions it is very simple, it will be very simplified. Now I have drawn some like lines here. What does this mean? That the portion above this line, the portion above this white line is lying in the cortex region, and the portion below the white line is lying in the medulla region of the kidney. Okay, understood? That is why when I had drawn the renal pyramid like this, I had drawn nephrons like this. Okay, the nephrons were like this. So the Bowman's capsule portion was in the cortex region, but loop of Henle lies into the medulla region. Okay, now this structure of nephron also varies. It does not mean that this will be exactly the structure of nephron and all of the nephron will be like this. The nephrons can be of two types depending upon the length of the loop of Henle. See, this is the loop of Henle starting from this portion. Okay, starting from this portion up to this portion. From where the PCT ends until where the DCT starts, this is the loop of Henle. Now its length can vary, and according to the variation in its length, it can be divided into two parts, or two different type of nephrons can be called depending upon the length of the loop of Henle. So what are those types of nephrons? Type of nephrons on the basis of what? On the basis of length of loop of Henle. It can be divided, or it can be categorized into either juxtamedullary nephrons or cortical nephrons. Now, by the name you can imagine this is juxtamedullary. Medulla is coming in its name, so it will go on to the loop of Henle will go on to the medulla region. That means if this was the pyramid, and here we had drawn the Bowman's capsule, and this is the medulla region, the loop of Henle will go long into the medulla region. So here we can say that this loop of Henle is longer, and this is cortical, means major portion of the nephron will be in the cortex region. So if this is the renal pyramid, this is the Bowman's capsule, so maximum portion will be in the cortex region, and here the loop of Henle will be short. It will go into the medulla region, that is for sure, but it will be shorter, and maximum portion of the nephron will be in the cortical region, and here the maximum portion will be in the medulla region, because the loop of Henle is very long. It can go up to this length also. Okay, now what are the percentage, like maybe that they are 50-50 in our kidney, or they are 10-90-10. So maximum portion, maximum number of nephrons are of cortical type. So about 80-85% of the nephrons are of cortical type, cortical nephrons, and only about 15% is the juxtamedullary nephrons. When we will study about the counter current mechanism and the conservation of water, then we will once again study about the differences in these, which is more beneficial to us and which is less beneficial to us. Okay, then we will study the counter current mechanism, then we will come to know about the benefits of having a long loop of Henle and a shorter loop of Henle. Okay, now let's start with the physiology of excretion, physiology of urine formation. Okay, how is the urine formed? Now first step is ultrafiltration. Okay, first step is called as ultrafiltration. The next step is called as reabsorption, and the third one is called as tubular secretion. Let's draw nephron once again. See, it's very easy. This is the PCT, this is the loop of Henle, this is the DCT portion, and here we come with the collecting duct. Okay, now collecting duct, why have I drawn some branches? Because collecting duct is not the part of one nephron. Okay, here other nephron, from other nephron some filtrate will come, and from here other nephron filtrate will come, and now they will be called as urine. So what happens is, okay, let's draw here the glomerulus. Now what happens is ultrafiltration. We have seen ultrafiltration, that is the filtrate is coming in the Bowman's capsule. What all filtrate will come? Amino acids, water, salts, in the form of ions. Okay, Na plus, Cl minus, all of the ions. Urea, very important. Then glucose. All these filtrate may come here. Now what is happening is, here we do have blood vessels, here we do have blood vessels. Okay, let's draw it very simplified, like this, in a very simplified manner. Okay, now from portions of PCT, from portions of loop of Henle, from portions of DCT, some things will be poured back into the blood. That is called as the reabsorption. The second part, reabsorption. Now what all will be poured back into the blood? All that what body needs at present or later after some time, which need not be eliminated. So body will need what all? Body will need glucose. And here we need to remember, or you need to focus, that all of the glucose and all of the amino acids, that means glucose and amino acids, 100% is reabsorbed. Okay, so if there is no diseased condition, then glucose and amino acid will not go into the urine, will not be found in the urine. So glucose and amino acids, all of them will be absorbed from PCT itself. It will not go up to DCT and then be absorbed. From PCT itself, all the glucose and all the amino acids, 100% will be absorbed. Now some amount of water may be absorbed, some amount of ions, salts may be absorbed. Okay, so these, some amount of urea also may be reabsorbed. Maybe body needs some amount of urea to maintain the osmolarity, so some amount of urea will also be reabsorbed. So out the total reabsorption that is taking place, where reabsorption will take place in the PCT, in the loop of Henle, in the DCT, and maybe it will also take place in the collecting duct. Okay, it can also take place in the collecting duct if needed, very minimal. So out of total reabsorption, about 70 to 80% takes place in the PCT. In the starting itself, whatever body needs, it will take. Now in the loop of Henle, this one, it's going down. Okay, when we trace it, it is going down, and from here it will go up like this. So this is called as the descending limb of loop of Henle, and this is called as the ascending limb of loop of Henle. Okay, limb of loop of Henle. Okay, because it is going down and it is going upwards. So the descending limb of loop of Henle is permeable, will allow the elimination of water. Okay, from here water will be reabsorbed easily. From the ascending limb of loop of Henle, water is not absorbed very easily. It will allow the reabsorption. Okay, it will allow the reabsorption of salts. The ascending limb of loop of Henle will allow the reabsorption of salts. So from here water will be reabsorbed, from here salts will be reabsorbed, from DCT also some amount of water and salt will be reabsorbed. Okay, depending upon the need, not depending upon depending upon the need, some salt, Na plus, Cl minus, some amount of water, some amount of urea will be reabsorbed, but major portion from PCT only. From collecting duct also it can be reabsorbed. Now coming on to the tubular secretion. What is tubular secretion? Now whatever we have reabsorbed, it may happen that body is thinking that now body is thinking that okay, we don't need that much amount of water. So a person has taken enough water, more than required, so the body will now eliminate it once again. So whatever has come into the blood, if it is not needed up to that extent, it will be again poured back into the filtrate, and maximum amount of tubular reabsorption is carried out in the DCT region under the influence of ADH hormone and aldosterone. Okay, ADH will allow the water to be put back. So the ADH will allow the water to be put back into the blood through reabsorption, and aldosterone will allow the salts or ions to be put back into the blood through reabsorption. So in the DCT portion, ADH and aldosterone will help in the reabsorption of salt and water when needed, but in the DCT portion, tubular secretion is the maximum. And tubular secretion of what? Tubular secretion means whatever blood does not need, it will pour again into the filtrate. So maximum is H plus ion. Okay, H plus ion and some amount of salt also, some amount of potassium. Okay, some amount of potassium, some amount of ammonia, because ammonia is also produced. Some amount of potassium, some amount of H plus ion, some amount of ammonia, some amount of urea. Okay, all this will be put back into the filtrate because now it needs to be eliminated. This is the last stage, and then finally when here whatever comes here, it will be called as urine. Okay, let's study counter current mechanism. Now this is the descending limb of loop of Henle, and this one is the ascending limb of loop of Henle. Vasa recta comes in opposite direction, so this is the descending limb of vasa recta. Okay, now what is vasa recta? This is the capillary, the network of capillary around the loop of Henle that we had drawn is called as the vasa recta. Okay, so this is vasa recta, and all the other portions which were around the PCT, around the DCT, and overall it is called as the peritubular capillary, but the specific portion which is around the loop of Henle is named as vasa recta. Okay, now this is the descending limb of of vasa recta, and this one is the ascending part or ascending loop of vasa recta. So it goes opposite. Now what happens is the filtrate is going in this direction. Okay, but the blood is coming in this direction. Now the osmolarity, what is osmolarity here? At this portion the osmolarity is about 300 milliosmol, then it will start increasing 300, 400, 600, 800, 1000, and at this portion it's 1200. Okay, so at the extreme end of the loop of Henle, or at the extreme end of the vasa recta, we can say at that portion it is about 1200 milliosmol. Our blood osmolarity is 300. Now why and how does this go on increasing? Increasing osmolarity means the solute amount has increased in the solution and the solvent is comparatively less. So the solution has become hypertonic, it has become very rich in solute. What happens is, we have studied that this portion, the descending limb of loop of Henle, will eliminate water very easily. So it keep on eliminating water. This is permeable to water, so it keeps on eliminating water. So when the water will be eliminated, so what all will be left? More amount of solute will be left. So by default we can understand that the osmolarity will go on increasing. That means the solute concentration will go on increasing. So that is why it has become 1200, right? Now when we go upwards in this direction, what happens when we are going in this direction? We can say that from 1200 it's going to, sorry, it's 1000. From 1200 it's going to 1000, then 800, 600, 400 and 300. So it's decreasing, the osmolarity is decreasing. Why? The water is not being added. Here the salt is being eliminated. Na plus and Cl minus is being eliminated from here. Okay, so that is why, and this will enter this portion, and then it will go on, and the water, and since Na plus is coming in this direction, water has come here, and from here water will come into the ascending part of vasa recta. So this is called as the counter current mechanism. Counter means opposite. So the filtrate in the loop of Henle is flowing in the opposite direction as compared to the blood in the vasa recta, and through the interaction between them, the osmolarity is being balanced. Okay, so this is called the counter current mechanism. So ultimately what happens, water is being conserved in the body because all of the water that has come in the interstitium, this is called the interstitium means the portion between the vasa recta and the loop of Henle, interstitium, middle portion. Here some amount of urea will also be present. Okay, so water is coming in the interstitium, and then it will ultimately come into the blood vessel, into the blood, it will be mixed. So water is being conserved. Okay, water is not being eliminated that much. So counter current mechanism helps in water conservation. So whenever there will be a scarcity of water in the body, maybe during fasting, water fast, so this will work, the counter current mechanism will work, and water will be conserved in the body. So that is why when we had studied about the juxtamedullary and the cortical nephrons, we had studied the juxtamedullary nephrons and the cortical nephrons. The juxtamedullary had long loop of Henle, the cortical had shorter loop of Henle. So if there will be long loop of Henle, there will be long vasa recta. So there will be long vasa recta, then more amount of water will be conserved in this as compared to this. So juxtamedullary, although it is 15% of the total, but it helps more in the conservation of water. Okay, so this is the significance of long loop of Henle. This is the importance of being having juxtamedullary nephrons and the cortical nephrons. This is the difference between their roles. Let's study about the regulation of kidney functions. So how are the kidney function, that is the urine formation, the osmolarity maintenance, that means the maintenance of salt and water balance in the body is done by the kidney. What all factors play important role. Okay, so first is the release of ADH. Now what is the full form of ADH? Antidiuretic factor, or it is also called as vasopressin. So this is a hormone. Okay, this is a hormone. It will act upon the DCT, acts on DCT, that is the distal convoluted tubule. It acts on DCT in antidiuretic. Diuresis means the removal of water. So it is antidiuretic, means it does not allow the removal of water from the body. So it will help in the conservation of water. Okay, in the body, because it is anti of diuretic. Diuretic means removal of water. Diuresis means more urine elimination. So anti of that means it will conserve water. So if it will conserve water, then it will lead to increase in the blood volume. Increase in the blood volume, therefore increase in the blood pressure. Increase means normalize. Okay, normalize. So whenever the blood pressure drops or the blood volume is decreased, then this will work. Okay, so this works when will it work? It will work to normalize the blood pressure. So when the blood pressure decreases or the blood volume drops, okay, then it will work to conserve water in the body. Okay, now it will also increase the osmolarity, right? It will also increase the osmolarity. The salt water balance will be maintained. Now, anti atrial natriuretic factor, that is the second portion, second factor, ANF, atrial natriuretic factor. What does it do and from where it is formed? Atrial, where is atria found? In our heart. So our heart forms this factor. Natriuretic, uretic in urine, in relation to urine, urea. Natrium, natrium means sodium ion. So what happens is when the blood pressure is too much high, okay, then this works because it is natriuretic. In urine you have to eliminate natrium, that is sodium. Heart makes this factor so as to eliminate large amount of sodium. And what was sodium doing? Sodium is responsible for increase in the blood pressure. So when sodium is eliminated through what? Through urine, because uretic comes here, through urine. So this is done, sodium is eliminated through urine. And what happens is, when will it work? When the blood pressure is very high. Blood pressure high means there is lots and lots amount of salt, lot amount of sodium, okay, hypertension. So when the sodium amount is high, then sodium will be eliminated. And we will study about the RAS. It will also suppress RAS. It suppresses the RAS mechanism. We will study about RAS. So it works in a dual function. It will eliminate sodium and bring down the blood pressure to normal when it is too much high, and it will also suppress RAS. Okay, so as to suppress the RAS will also act to increase the blood pressure and the blood volume, as it was doing in the case of, as the ADH was doing. Okay, so ADH and RAS will work similarly to increase the blood pressure and blood volume, but ANF will decrease it. It will work in opposition to the RAS and ADH. So RAS is the renin angiotensin aldosterone system. Three things are coming: renin, angiotensin, and aldosterone. Now we'll study one by one. Renin. This is produced by the juxtaglomerular cells of the kidney. Where are these juxtaglomerular cells formed? Renin is formed by JG cells. Okay, so what are these JG cells? These JG cells are found in the afferent arteriole. So let's imagine this is the afferent arteriole portion, and here we have our Bowman's capsule, and here somewhere here we will have our DCT portion, a portion of DCT. Now there are some cells here, okay, let's change it. Yeah, this is the afferent one, okay, afferent one, and this is the efferent one. This is the DCT, this is the afferent portion, and here is the Bowman's capsule. So these are the JG cells, and this is called, and here are some cells of Lecci. So this 1, 2 and 3, 1 plus 2 plus 3, they make the juxtaglomerular apparatus. Okay, now this JG cell will release renin when the blood pressure falls below average, blood volume is very less. Okay, what happens is renin is secreted. Now this renin will convert angiotensin which comes from liver. Angiotensin, this is going on in the blood. Okay, blood vessel. In the blood vessel, this whole thing is going on in the blood vessel. Angiotensin will be converted into angiotensin 1 and then angiotensin 2. This is a powerful vasoconstrictor. So what it will do? Since it is a powerful vasoconstrictor, it will constrict the blood vessel, which will lead to blood pressure increase. It will also lead to release of ADH, which will help in water conservation. Then blood volume will increase, and it will also lead to release of aldosterone, which will lead to salt, or we can say sodium ion conservation. Again, when sodium ion conservation will be there, blood pressure will increase. So our problem area has been solved. The blood pressure and the blood volume was dropping below average. It has been solved. The blood pressure has been increased by constructing the blood vessel, blood volume by conservation of water, and again through aldosterone, which will, these two will act on the DCT. Okay, both will act on the DCT, distal convoluted tubule, and then salt will be conserved, which will again lead to the increase in the blood pressure. So these are the how the regulation is done of salt and water balance, how the balance is maintained. Now let's go on to the micturition. What is micturition? Micturition means voiding of the urine. Okay, simple, voiding of urine. That means eliminating the urine. Our urine is of slightly acidic, means pH is less than equal to 6. Its color is pale yellow, okay, due to a certain pigment called as urochrome. It contains urea, and about 20 to 30 gram of urea is eliminated per day in our body. Approx 1 to 1.5 liters of urine is excreted out of a normal human being from the body. It is a neuromuscular mechanism or neuromuscular reflex, we can say that, because sphincters are muscles, right? So sphincters, okay, we can say that it is a mechanism which is controlled by nervous system. Okay, we can say that it is controlled by nervous system, not neuromuscular, but it is controlled by nervous system. So neural reflex, we can say that neural reflex. Okay, now what happens is when the urinary bladder, for example, this is the urinary bladder, here the ureter, the urinary bladder is filling. So there are certain cells which is called as the transitional epithelium or urothelium in the ureter, right? They are stretchable, they allow stretching. And here also in the urinary bladder, certain muscles are present called as detrusor muscle. Now, this detrusal muscle is smooth muscle. Okay, it is involuntary. This is urethra. Urethra is lined by two sphincters. This is the internal sphincter. Okay, internal sphincter. And then here we have external sphincter. Now, both of them are neither voluntary nor involuntary. The internal one is involuntary, but the external is voluntary. But in children, it cannot be voluntarily controlled. So children, so the up to 1 years of age, they cannot control the urge of urinating. Whenever their bladder is filled, they will urinate, no matter what. Okay, but we can control because our external sphincter is voluntary, but internal one is involuntary. Okay, let's talk about some diseases. Now, uremia means blood urea level has increased beyond normal. Okay, so there's a lot of urea accumulated in the blood because the kidney is not working properly. Renal calculi means there are stones of either calcium or oxalate has been deposited, so they are called as the kidney stones. Glomerulonephritis means inflammation has been taken place in the glomerulus, so inflammation of glomerulus. These are certain disease. Then certain disorders, glycosuria means when glucose is found in urine, which is not a normal condition. Hematuria means if traces blood in urine is found, it is also not a normal condition. Blood should not, RBC, blood cells should not be found in the urine. RBC, WBC, they should not be found in the urine. If it comes, then it is called as the hematuria condition. Now, the last portion is the hemodialysis. Now, what is hemodialysis and when it is done? Hemodialysis is done when the kidney is not functioning properly. Kidney is not functioning properly or failure of functioning of kidney. What happens is when the patient is taken to the hemodialysis, there is a box, there is a machine which has certain fluid in it, and this fluid has exactly same constituents as of our blood plasma. There is a cellophane tube through which the blood passes. Okay, now through this tube the blood passes. The patient's from the patient's artery. Okay, from the patient's artery, the blood is taken into this cellophane tube, and there is the blood and the plasma constituents are exactly same, but except this does not have any urea is absent, but here urea is present. So urea will come into this dialyzing fluid, and then whatever purified blood which is free from urea will be returned into the vein of the patient. Okay, so vein, why? Because here the blood pressure is low, and artery, why? Because here the blood pressure is high. We cannot return into the artery because the blood pressure is too much high. So this is the hemodialysis. Now, one more portion, last portion, is the role of other organs in excretion of various waste materials. Now, what other various waste materials? First, through lungs, what we excrete out? Carbon dioxide and some water vapors. Okay. Then through liver, through the digestive waste, bilirubin, biliverdin, some amount of vitamins, drugs, all these are excreted out through liver, produced by liver and then excreted out through the digestive waste. Some amount of waste is excreted out through sweat glands like oil. Some is excreted out through sweat glands, some salt and water, sebaceous glands, some oils and waxes, and our saliva also excretes some amount of nitrogenous waste. Okay, so these are the organs that also help in excretion up to some extent. Majorly it's excreted out by our kidney. So this is all for this complete chapter, which has been covered in one shot. Thank you very much for listening and having patience.